it is really my honor to be invited to such a big event, 5th and 40th anniversary. Today I choose my talk title as implantation model to study in vitro. But as a person who spent the whole year of my research on oocyte maturation, embryo development and implantation area is a little bit out of my field. So it was a little bit nervous stage today. But I'm so glad we have Dr. Jung before me. And I would like to introduce my system we developed lately. Oocyte maturation, I've been focused on early stage of folliculogenesis and oocyte maturation. And having a good quality of oocyte is really important for the afterwards process, such as fertilization and pre-implantation embryo development, as well as the develop to the good quality of blastocyst for implantation success. The year near 2000, it was like a boom for finding the DGs and biomarkers in every system. So at the time, I was lucky to have enough funds and manpower to do things like that. And so I focused on folliculogenesis, subtractive hybridization, and ACP-PCR gene fixing method for oocyte maturation, as well as microarray system for early folliculogenesis and implantation sites. From this time, implantation was my interest, but I didn't have enough time and grant for study implantation. So today I would like to just briefly touch my research on gene fixing, what kind of genes I found and what I did during last decades, and in the second half, I would like to touch the implantation story. Oocyte is really interesting and beautiful cell I met, and it has very unique cell cycle, twice of arrest. From the birth, oocytes are arrested already at GV stage, and after the ovulation, it is maturation is resumed, and again stop the cell cycle at M2 stage, waiting the sperm to fertilize. So, but it is really big, because it has to in charge for fertilization and embryo pre-implantation, embryo development, such a thing. So I was interested what kind of genes are important for oocyte maturation and what kind of genes are important for the fertilization, things like that. So we applied ACP-PCR technique at the time. It has 120 primers for PCR, and by using that specific PCR primer, we can get the GV specific or M2 specific or selective. That means big change in expression. So by using 120 primers, we found 4 GV specific, 97 GV selective, 26 M2 selective, and nothing for M2 specific gene. From that time, if students come to my lab, and they can find out their interesting genes, and we did the loss of function study from that time. I was lucky having the RNA interference technique from year 2002. because I couldn't have the knockout animal technique. So I found many target genes, but I didn't have tool to study its function. But by having RNA interference technique, it is efficient and specific, fast, inexpensive, and as well as it has high success rate to study loss of function. By having double-strand RNA injecting into the cell, we have specific knockdown of the target genes in the IBMX media, and without that gene, we can do oocyte maturation. Then we can observe what has happened in that oocyte without that genes. So we observed the morphological changes, cellular changes, and other cytoplasmic changes. To confirm cytoplasmic changes, we did in IBF and the other processes further. So I would like to show some kind of videos of the normal oocyte maturation process, and then without other genes, what happened to oocyte maturation. I couldn't make the same slide here, so I had to separate. It is normal oocyte maturation process for overnight. You can see the cytoplasmic streaming, very active, and then polar bodies are beautifully, cytokinesis is done, and polar bodies come out. So almost 100% of oocyte maturation occurred. It is mouse oocytes. And then, without OBOX4, it is other gene we found. Without OBOX4, you can recognize, at 24 hours, at the normal time we observed, it seems like it's M2 without polar bodies. However, we realized that it is polar bodies coming, but cytokinesis was incomplete and polar body was gone. So it is the cell division, first the cell division didn't occur. It was really interesting observation. So with video recording, we realized that many things happened during the process. And then without TXNIP, it is an enzyme in PPT pathway other than glycolysis. You can see without TXNIP, the cytoplasmic streaming is gone, and the maturation didn't occur. So it is important for maturation in OBOX4 too. But in the case of GAS6, without GAS6, nuclear maturation is occurred. So it looks fine as like a normal. However, without GAS6, what happened was, without GAS6, it looks like normal, but GAS6 is gone. mRNA and protein didn't. So without GAS6, what happened was, it is not normal. Cytoplasmic maturation didn't occur. So sperm penetrated in the cytoplasm, has swallowed for the zygotic formation, but pronuclear formation was observed in control and sham control. However, GAS6 RNA interfered in GAS6 knockdown oocytes. Sperm head doesn't have decondensation. Sperm heads there. So and the the cortical granule exocytosis also didn't occur without Cas6. So Dr. Kim at the time found that without Cas6, the MPF activity is decreased, and calcium oscillation also couldn't was normal, abnormal. So pronuclear formation didn't occur, and she came back after 2 years of her postdoc, other laboratory. She came back and continued the Cas6 experiment. And she found that what happened in the cytoplasm, she focused that, and then found that normal mitochondrial activity and normal glutathione production and heparan sulfate production was not normal without Cas6. And this is really important for normal fertilization as well as for the SCNT, nuclear reprogramming study. The normal cytoplasm is required. And she focused further for mitochondria, what happened to mitochondria. And she found that the mitophagy regulation was controlled by Cas6, and it is reciprocal. So due to her efforts in Cas6 area, we find that it is really important gene for cytoplasm maturation as well as fertilization and further the embryo development. Lately, she decided to do the recovery experiment. So she injected Cas6, the recombinant Cas6 into the mouse cytoplasm and recovered all the dysfunctions in the mitochondria and other cytoplasmic situation. And it is important because aged oocytes from advanced age woman, the oocytes also having deterioration in the cytoplasm. So by having this kind of therapeutic injection of the target genes could recover the aging. However, the oocyte is also very important for as a germ cell. So it is experimental right now at this moment. So by having many cells, and we realized that a couple of genes are important for M1, M2 transition for nuclear maturation, but some genes are important for not important for nuclear maturation, but the cytoplasmic maturation and further embryo development. And we learned that there is a maternal effect genes that is important for embryo development. And we added CBOX and Cas6 as I didn't have story here, but CBOX and Lin28 is a gene. Without this gene, embryos cannot develop up to 2 cell to 4 cell. And as I introduced Cas6, without Cas6, oocytes cannot develop as a zygotic site. So we added up the couple of MEGs almost 10 years ago, and I Googled how many MEGs I reported these days, and I found it was 82 lately. So science really developed. Come back to the story of implantation. Long time ago, when we did microarray, we had chance to have laser capital micro dissection. This is the technique which has from the paraffin or cryosection, we can isolate by using laser, we can isolate the target area as much as small. So in here, we focused with the implantation site in blue and intersite without embryos. So we wanted epithelial cells from implantation site and intersite. We captured that epithelium with LCM, and we did microarray between these luminal epithelial cells from IM and inter sites. And we found many differentially expressed genes, and among them, a couple of are related in signal transduction, ECM structures, things like that. And also they are related with calcium regulator. So at the time, we, by having the software, the finding that these genes are important in implantation, as well as calcium is important for implantation. But as I said, I didn't have any tool to study the gene function at the time related to implantation. So I said, Dr. Jung, IMBU have that kind of technique. So come back to the story, the novel artificial endometrium model. A couple of years ago, by having one young fellow professor in biomaterial area, and Dr. Jeon Young-pil in Seongshin University, and Dr. Park Seok-hee in Busan University, we had opportunity to work together for endometrium, making the scaffold to study implantation in vitro. So we applied the grant from the Ministry in Health and Welfare. We got the grant to do this, and we established to making the platform for implantation study. But however, the reason why I have this topic today was, our paper is flying from biomaterial area as well as reproductive area, because I guess we have 50 and 50% content. Biomaterial ping-ponged our manuscript to reproductive journal, and reproductive journal ping-ponged to biomaterial. So I was, I lost myself. What should I do? So I would like to get your opinion about our story and suggest a good journal to apply again. However, it's under review at this moment at the Biomaterial Journal. Now, so to meet the two unmet needs at the time, we interested about making scaffold to study in vitro for implantation as well as for ovarian function to study the early follicular development. So in this part, we wanted to develop a 3D biomimetic culture system closely mimics the human endometrium. So by having Professor Park at Busan National University, he had a technique, 3D printing, electrospinning, and bioprinting. By using this kind of technique, he made the artificial endometrium scaffold for us. Then we used the stromal cells and epithelial cells with these 3D conditions. It isn't easy as much as we thought at the first. During the time, there was a review about the embryo implantation study in the lab. The Institute of Bioengineering of Catalonia, it is from Spain, and I guess they are focusing their research in the implantation between cell-cell communication of embryo and epithelial somatic stromal cells. And I will introduce the recent paper by this group, and the third author was primary author for the recent paper. They watched real embryo growth. They made the video embryo growth in vitro. Anyhow, with this review, we can recognize that there's lots of cells used for endometrial part, epithelial cells and stromal cells, and also it is very difficult to get human embryo, but so there's many cells are used as embryo surrogates. Trophoblast cell lines are used to make the embryo spheroids. So we accepted JA cell and Ishikawa cells, Ishikawa cell, because Ishikawa cell has receptivity for embryo implantation. So there's lots of 2D co-culture and 3D things here, but we decided having D structure. But we focused, we wanted one more addition of basement membrane. So rather than having just co-culture of two cell populations, we wanted stromal cell, we wanted to add basal membrane between stromal cells and epithelial cells. So we asked Professor Bach to make network, mesh on the top of stromal portion. So he tried many concentrations of the biomaterials, and finally he succeeded to have mesh as a basement membrane structure. Then we add up stromal cells below the mesh, and then attach the epithelial cells beyond the basement membrane structure. Then JA cells are added up to study the cell-cell communication here. So it wasn't easy, as I said. There's lots of try and error using the different structure, different components, and at first we had glass to see the development, but glass structure is difficult is confronting the cryosection after the culture. So we changed the glass bottom and the height and the sectors. We tried and the holes and the side to inject stromal cells with fibrin. Fibrinogen with thrombin. Without this one, we cannot even inject the stromal cells into the structure. So afterwards, we decided to combine these two factors with stromal cells. And then third time, we removed the hole, and again we had hole, and we changed the heights to, and we changed the bottom, things like that. So finally, we've got the structure, very simple. And we have PCL structure with PCL nanofiber network as a basement membrane. And we have PET membrane to look through the structure process under the microscope. So this is the simple scaffold, and this one is stromal cells filled in fibrin gels under the network mesh. network, and then added the Ishikawa cells on top of the mesh. So by having that final decided scaffold, the spheroids were added. We have EGF expressing JA spheroids to compare to the separate the trophoblast cells out of the endometrial epithelial cells. So by having EGF expression, so we compare the cell culture condition on that the basement membrane here. We had Ishikawa cells as receptive cells, and negative control as a negative control. AN3CA cells are non-receptive epithelial cells, so it was negative control. And in the fibrin gel itself, these AN3 cells, the distribution wasn't even, and however, on the mesh, the basement membrane structure, their culture condition was very even and healthy. And even after 7 days of long culture, long-term culture, there's the cells are alive, and there's not many dead cells. So it is good. We confirmed it is good condition. And then after the culture of both cells and together, Dr. Kim, got this good picture of the well-attached on the mesh, the JA spheroids. For attachment assay, as we expected, it is better than glass itself, and fibrin gel was in between of the artificial endometrium, showed very good success rate in attachment. But even in non-receptive AN3CA cells, there's some attachment, but it's lower than Ishikawa cell. And also invasion assay had the same result. The artificial endometrium with the two cell, somatic cell, stromal cells and epithelial cells, and in between the ECM-like basement membrane, we had very good invasion in the system. So after the culture, she did RNA-seq to see what happened between cells. So it is initial time cell, initial points, and then after the cell-to-cell communication for 24 hours, she separated Ishikawa and spheroids and AN3 and spheroids. So compared their gene expressions. And you know, myself, I'm so simple person. So I thought, you know, AN3C, I mean the non-receptive one, there will be nothing happen. But the gene expression was kind of active than Ishikawa cells here. So we verified some genes by using the PCR. So chip assay records are rare. Then we did string network analysis to see the DG's dynamics. So this is Ishikawa cell itself, the endometrial epithelial cells, and this is spheroid cells on the Ishikawa cells. So we can see some genes are working on related angiogenesis and some cell migration things, and so it looks fine for as a scaffold to study in vitro. And And also, we found that rather than epithelial cells, spheroid itself has more dynamic changes there. And as I said, on the non-receptive, the epithelial cells, there's some more, it seems like more dynamic changes occurred. However, the genes are not important for direct invasion things. However, it is just a spheroids, so it will be different story if we have real human primary cells and the human embryos on top of. So it is a good system for personalized study for any infertile patients. So we were lucky to get a couple of rare endometrium samples from NOMAR and for infertile RIF patients. It was 7 samples from the patients. So this upper layer shows the, so everything is the same. Chromatic, stromal cells are the cell lines, but epithelial cells is from patients. So it is NOMAR, epithelial cells from NOMAR, and epithelial cells from RIF patients. And this is jar spheroids. So you can see in the normal epithelium, the jar spheroids had intact opposition and attachment with compact texture. However, on the epithelial cells from RIF group, you can recognize there's something different. The jar spheroids has very loose communication, as well as its structure is really, a little bit far different to the normal, the spheroids on the normal epithelial cells. I was kind of excited to see. Looks like it's embryo on top of the epithelium. But so if we had enough time and the opportunity to get the more samples, it would be good to have this kind of system to study patient by patient story to see what is happened to here. But unfortunately, at the time, the grant is, the period is gone, and the people who worked with us graduated, and the clinician left, Dr. Lee left. So we have only the anatomical structure here, but however, I would like to introduce this system to the people to study the implantation, because for in clinical fields, the artificial the system such as this ECM mimicking, having the ECM mimicking co-culture system will be good for the study to patient story. We successfully co-cultured stromal cells and epithelial cells with trophoblast spheroids to study attachment and invasion here. For gene expression analysis, the sample wasn't enough to do single RNA-seq for patient story, but it's again a couple of years ago. So technique for single cell, the analysis will be more than sensitive to have small amounts can be enough for the molecular study nowadays, I guess. So the potential application of this, the 3D structure is good for study implantation biology, uterine disorder, and microbiome study, things like that, for personalized study to improve IVF outcomes. So this is the end of my story, and as I said to introduce, the paper came out last August. The group from the Spain, I don't know it works or not, internet. Here. It is, I mean, the mode, and it worked for me at home, but anyhow, they had very short clip, a couple of seconds, not seconds, milliseconds. The embryos dip into the collagen. This is collagen gel, and the embryos growing in the collagen gel. So I guess it is first time to see real embryo growth outside. To acknowledge this presentation, Kim Kyung-hwa in my laboratory spent her 20 years with me for all the gene fishing story, as well as the scaffold structure study for today. And Park Seok-ki, Professor Park showed his opinion that anybody wants to study biomaterial in this field, he's open for collaboration. So you can touch with him. And due to Professor Chun Young-pil, he gave me the opportunity to work together with biomaterial person. As Executive Director of Child Fertility Center, at the end of my talk, I just briefly introduce our institute. As you all know, Louise Brown was born in 1978. She is the first test tube baby worldwide, and we celebrate in 2017 in ASRM her 40 years of birthday. And we are celebrating 40 years of ART in Korea, so I thought it is good time to show her. And Cha University Global IVF Group has other hospitals through the world, but in terms of IVF clinic, we have 6 centers in Korea, and one in Japan may be open at the end of this year, and one in Singapore, as well as we have a couple in Australia. And in Korea, we have 6 right now, but in November, we'll open in 7th center in Magok this year, and we are working around 500 employees together, and 84 and 200 embryologists together nowadays. It's a couple of months ago. And for Australia, we joined in 2018, and right now we have 13 clinics in 5 cities and 14 counseling offices through the Australia. And this is the slide I would like to show you. And I found out the success rates of clinical pregnancy rate in 2022. It was SART data and Singapore national data. So it was the clinical pregnancy rate. So they have a live birth rate in Singapore and United States. But in 22, C Hospital in Korea, we are, its average success rates in 50, and in Daegu and Seoul Station, we are having 60 and 70% of success clinical pregnancy rate lately. So I would like to say that the clinicians and biologists in Cha Fertility Center are working together to improve the implantation rates as well as the the finding out new and safe way to improve the clinical pregnancy rates. Thank you for listening my talk. Thank you.